Sometimes they divide RNA up between what they call coding RNA and non-coding RNA. Coding RNA, what would be an example when you say something's coding? Codes for a gene, right? So we're talking mRNA. Whereas the non-coding RNAs can be things like tRNAs, rRNAs, microRNAs, small nucleolar RNAs, all those kinds are considered non-coding RNAs. Okay. But yes, they don't encode for a gene specifically. Alright, so now we're going to go over some of the roles of the different RNAs. Now for this exam, we are not going to be covering chapter 9, so it's only, 5, 6, 7, and 8 it's going to be on this exam. But chapter 9 will not be on the exam. And this is really a review of tRNA here, because tRNA, and you should know its role in protein synthesis. What role does it play in protein synthesis? What does a tRNA do? It responds to the codon through the anticodon. It brings the corresponding amino acid. And I like this figure here because it gives you a rough idea of the size of the amino acid. The amino acids themselves are really tiny, especially if you looked at, this one's not drawn to scale, but if you looked at tRNA drawn to scale, and there's a great video that's on the the link to it was on the PowerPoint that would show, and I'll try to have it on Friday's class, that shows the actual, based off of crystal structure, the actual process of protein synthesis. And it's amazing how tiny the amino acids are compared to the RNAs that are involved. And so you're supposed to know the names of the different parts of the RNA, the tRNA here, and this is the amino acid, and so this is called the amino-acyl stem. And remember, the enzyme that's involved is called, if it's not, the CCA wasn't already there, it's called the tRNA nucleotide transferase, which is why it adds that CCA onto the gene, onto the RNA, in order for it to be amino acylated, otherwise it wouldn't work. And it's really proficient at discerning the sequence and putting the correct, amino acid where it's supposed to go. And then there are many different types of tRNA, many different types. And so you have to have a minimum of at least one per amino acid. And some amino acids have multiple tRNAs based off of that genetic code and the redundancy in the genetic code. And remember, tRNAs can be they're highly modified. I just went over some of the examples in the previous class. This is mRNA. Oh, and tRNAs are medium-sized. I mean, it really depends. Like I think it's one of the phenylalanine tRNAs had that huge variable loop, but they're relatively medium-sized. mRNAs can be really large. Okay, they can be variable in size, and it's a direct transcript of DNA. It's what contains the codons. It also contains introns and exons for those organisms that do that encode those. I'm not going to go into very much detail with mRNA because the fact is that's something that you guys should have covered already in biology and genetics and cell and molecular and all these other classes. So that's why I want to focus more on some of the other RNAs. This is the rRNA, or ribosomal RNA, and it's large. Whenever they come together, it's going to be large, and it is truly a ribozyme in the sense that the RNA components are what is involved in the chemistry of the reaction. Okay, the proteins are ancillary to that, and there's very, very few types of rRNA. tRNA, there's lots of types. mRNA, I mean, there's lots of ways to splice mRNA and different genes and all that. But rRNA, there's very little heterogeneity because the fact is it has to do a certain function. If you have a lot of heterogeneity to that, you could lose the function of the ribosome and then you're dead. And so there's just not a lot of heterogeneity with them. Okay, this is from the prokaryotic. They've been able to crystallize, and at the time that it was crystallized, this was the largest complex that was crystallized, and it's kind of hard to see here. But there are two subunits that they call the 50S and the 30S subunits in bacteria. Does anyone know what the S stands for? Svedborg, and it's a horrible, horrible way of characterizing. They used to use it for mass, but it's not really mass. It's based off of buoyancy. And so that's why you may have seen where they would have the 50s subunit and the 30s subunit, and when they come together, they make the 70s subunit. Like, the math does not add up. And so, I don't know, there's a few RNAs and stuff that they still link that way, and they still call, instead of saying kilodaltons or anything like that, which that's mass, molecular weight is in daltons and kilodaltons. There are some that they still call like 5s and 50s and 30s, and when they come together it makes a 70s. And that's based off of centrifugation and buoyancy. And so the old-fashioned way they did it, shape can affect how buoyant or how dense it is. And so the shape, not only the mass, but the shape and the way that's how you can have two things that weigh a certain amount come together and they apparently don't weigh the same, And the reason why is because it changes its shape, and so it has a different buoyancy. And so it's just a historical artifact now. But there's a 50s and there's a 30s subunit. And the tRNA actually binds in between. And the lighter color here, this lighter color, that's RNA. And this darker color you only see on the outskirts, the periphery, those are proteins. Those are really tiny proteins, but you see this really large RNA subunits. And so that's why it's primarily RNA. And then these that are 3 different colored ones right there, that's tRNA. And then there are, depending upon the books that you use, they argue about whether or not that there are 2 versus 3 tRNA binding sites. Okay, some of them call them APE, some of them call them APO binding sites. And we'll go over that in just a little bit. But the tRNA binding sites, which I usually do refer to as having 3, is at that interface between the 2 subunits. And so it's not active as just a 50s or just a 30s. It becomes activated whenever the 2 come together to form the 70s. Okay, and so this is the ribosome structure, and this is when it's closed, and you can't really see where the tRNAs are binding and everything because it looks like a little chute. It comes down and it goes, in and out. However, you crack it open like a nut, then you'd be able to see like where the tRNAs are bound. Okay, and so that's why, once again, we can see the RNAs is predominantly RNA-based, and you just a handful of these little tiny proteins along the outside, or the periphery of the complex itself. Okay, this on the PowerPoint, this is where you could actually click on it, and it would take you to what used to be a movie. I haven't checked to make sure that the YouTube video is still available, but it goes through the various parts. Just check something out, right? Okay, and so I do want you to know the 3 binding sites and how this all happens. And we're not going to go into as great a detail as you probably went through in Cell-Malek. But what happens here is that you first have initiation factors that start it, then you have elongation, and then finally you have termination. And so the very first, what is the first amino acid for that starts translation? It's FMET. And what does the F stand for? It's formal, and not formal as in a tuxedo. Formal as in FORMYL, and that's the old term for an acyl group. And so, let me do it this way. This is a formal group. It's just an aldehyde. Okay, and it gets added on to the methionine, and that's why it's called FMET. It's a little different. It's not just your regular methionine. It has this extra aldehyde on it. Okay. Now, many times later on that gets cut off because, the proteins themselves get modified later on. But, so not every protein, when you're looking up the actual structure of it, when it's fully modified, will start with methionine, but many of them do. And so that's that AUG. Let me blow this up so we can see a little bit better. That's the AUG codon. So the initiation factors will bring the F-met there. Okay? And there are 3 binding sites. And this is going to enter into the first binding site, which is called the A site. And so I remember it's spelled APE. A-P-E. Let me move this up. Well, maybe not. There we go. Okay. So then what happens is it moves over for the elongation to the P site. Okay, and the P is for peptide synthesis or peptidal transfer group. And so then what happens is the A site is then, sorry about that. The A site is then open so that way the next tRNA brings in the next amino acid in the A site. And the A site is for amino acylation because it's the site that's bringing in the new amino group, what will be on the amino group. Okay. And then peptide synthesis will occur. Okay, and so it's just a simple relatively simple nucleophilic attack where one amino acid will attack the next amino acid over, and in doing so, the original amino acid, or where the peptide had been in the P site, is now moved over to the amino acyl site. Then what happens is it just switches and it moves over to the P site. The site that is not listed here, because this one just shows 2, but this one right here is called the exit site, or the E site. For some organisms in some books, they'll talk about having an E site. And that's just where the used tRNA is there just before it gets kicked out. Okay, and so that's why I remembered it spelled APE, A-P-E. Of course, this one's got EPA, but you look at it from the other direction, it's spelled APE. And so then the next amino acid comes into the A site, then the growing nascent chain attacks it, gets moved over to the A site, and everything goes switched to the P site, and you get the other amino acid is kicked off out the exit. Okay, and so when this happens very fast, and it's very proficient, and it is not promiscuous in that sense, so you don't want a lot of mistakes being made. And the growing chain, the chain really does start to grow and can get quite large depending upon what protein is being made. Okay, I'm wondering if I. Okay, and so that described a lot of the tRNAs and the rRNAs, and even the mRNA, which was what was being read at that time. Now we're going to move on to some of the smaller non-coding RNAs. So we're talking about microRNAs and small interfering RNAs, which many times they get used interchangeably. Okay, so these are very small, usually around 20 to 25 nucleotides long. They do not encode for a specific gene, right? They don't make a protein. That's why they're called non-coding. And many times they're involved, since one of the reasons why sometimes they used to be called silencing RNA is because they're involved in gene silencing and gene regulation. And the little pictorial that I have on the other side is just one specific cascade. They're involved in cascades. So some of them are even involved in things like apoptosis, which is the reason why there's a possible role in cancer and cell proliferation. Some of these are involved in letting other enzymes and stuff know where RNA should be modified at. Because you've got to specifically modify, for example, that one tRNA in that one spot has got to have the dihydrouridine. Well, thank you. And so some of these are actually involved that way because what they'll do is, since they can be complementary, they will bind to the target RNA sequence, and then wherever they end, we'll let the enzyme know, okay, this is where I've got to modify and add a methyl group or this kind of functional group or take off a functional group or something like that. Okay, and so this is just one example. Many of these microRNAs are also, just like tRNA and mRNA for that matter, they're made as a precursor form, and then they're modified, they're cut at specific spots. And that way it just helps with regulation, because you don't want any of these to go out of whack. You always want to have nice control points. So all of these have controls. And so this is just one example here on this side. This is the Bantam RNA. This is just one example of a microRNA. And it's made as a precursor. And so then there are enzymes that come in, they will cut it and trim it down to the correct size. Then we have things like dicer is one of these examples of protein complexes that will recognize these. And many times we'll even split them apart to where it's now single-stranded. But then they are analogous to other regions of RNA or DNA. They can bind to that complementary sequence, and that allows for other complexes to come in and cut and cleave that. Once it's cleaved, that gene would then be silenced. It's been cut out. And so this is one way that they're allowed. And so the RISC stood for, I think it's RNA interference silencing complex. I may have the right idea, but the wrong, what the letters actually stand for. But it's where RNA has interfered with it, and that's what causes the gene silencing to occur.
So what silences genes through transcription?
Pardon?
So what silences the genes?
Right, because it really will, yes, because it can literally cut it out. That's why a lot of times with dicer and some of them, they show them as little scissors. Because what happens is if it's complementary to that sequence, then it binds to it, and then now it's no longer single-stranded, it's double-stranded, and this double-stranded complex is what then allows for the endonucleases to come and cut it. Okay, let's see what's next. Here's a little picture. This is the dicer picture, which was taken from the older book. And so you can see it's a really, really huge complex. And what they've done here is they're cutting the precursor, and then through ATP, this RISC, other proteins come, they separate it across to where now it's only single-stranded, and then that's complementary to whatever the target gene is. And the target gene then gets cut. Whoops. So before we get to the snurps and snorps and all that kind of stuff, I'm going to go ahead and end class with prayer. And on Friday,